right, that's going to be a tough act to follow. So hopefully my slides that don't have any memes or GIFs will also keep your attention. But I do have this pretty picture from Manus Biosystems. So this is their Augusta, Georgia sweetener facility. It's one of the known aerobic producing bioprocesses that's out there. They bought this from NutraSweet a number of years ago. And so what I'd love to see is the CABBI slides evolve again to have one that's representative of this next generation. Can we industrialize our bioproducts? And so most of you have probably been into the labs to see the small scale bioreactors that are down there in the lower right. You've probably been over to the IBRL to see the pilot scale facilities. And where we want to go next is to commercial production. And so if you see back here, I guess I don't have a pointer up here, but if you look in the kind of spiral just to the southeast of the I in CABBI, that's a 5-story tall airlift reactor. That is what Manus is going to use for production of their various sweeteners. They have the same types of bioprocess units that are in the IBRL in the white building that has the label Manus on it, and they have that on different floors. That building is also about 5 stories tall. So if you can't tell, I'm going to be talking about some differences in sizes, and this is a perspective that I learned while I was on sabbatical. Thanks to the Alexander von Humboldt Foundation, I got to spend almost a year in Germany with Ralph Tekors, who's formerly of Evolva. He had helped develop their tryptophan production processes and is now a well-known bioprocessing expert. And so Will Cordell, Dr. Will Cordell now, who was a CABBI student, came with me for a period of time and got to work on some projects there in the Tekors lab. And then Shufeng Wang, who's here, is now taking on a lot of this work inside CABBI. So what am I talking about? Process scale-up. We're taking things from the Petri dish, the microtiter plate, up to the test tube, up to the shake flask, to the 1 and 10 liter bioreactors, to the pilot scale that are at the 1,000 liter reactors, up to those 5-story tall things outside in an industrial plant. And so what's different about those different vessels or things that we're using to do the same biology or the same chemistries? Well, just the physical scale of them, the amount of material that you have, that's going to change a lot of the chemical properties. The physical properties, the pressures, the other mixing conditions that I'll spend a lot of time talking about, those are very different. And then those two things, the chemical and physical differences, manifest themselves onto the organisms and complicate their life cycle. And so the lessons that I want to leave you today is how these organisms that we're studying and we're optimizing in the laboratory now need to go through another re-optimization if we're going to successfully transfer them into industry. And so just to kind of give you some rough numbers, right? A lab scale 1 liter reactor, right, is a very small system. Pilot scale reactors that we have, they're probably about on the order of 1 meter cubed. Those we can operate almost identically to the laboratory. And so we can get great mixing, we can get great performance. There's not a lot of hydrostatic pressure on those vessels, and so they can be thought to be fairly homogeneous. But when we move to the larger scales, you'll start to see that there is a huge pressure shift, up to 5 atmospheres at the bottom, if you think about just the weight of the water. And so that change in pressure at the bottom versus the top of the vessel, which is going to be atmospheric, is going to shift all of your concentration solubilities and change the way that chemicals are going to dissolve into water or leave water and change the local environments pretty dramatically. The other thing to remember about these types of processes at large scale is you can't just do the same types of additions and simple manipulations that you can in the laboratory. So if you think about just creating the media, how do you mix all of the materials that you're going to want for a 5 meter cubed, or sorry, for a 100 meter cubed vessel? Think about the amount of sugar that you would need, and just I used round numbers of 100 grams per liter. Right, you can scoop a bunch of that for a small 1 liter reactor, but you would need 2,000 pallets of your normal Domino sugar bags in order to do the same thing for one of those large scale reactors. So just the physical manipulation is also completely different. But this talk, I think, is going to focus mostly on the challenges that we face, the organisms are facing inside these large bioreactors. So there aren't that many of these in the world, and they're certainly not instrumented in the way that our laboratory facilities are. So we don't know exactly what the concentrations are inside these systems, so we turn to modeling. And so this is computational fluid dynamics modeling of adding a particular substance at the surface of this tank and then watching it distribute itself through the entire column. And so this is typically defined as a characteristic mixing time when that concentration becomes nearly homogeneous. And so we've got correlations that would say, okay, for given stirring, for certain geometries, this is what we would expect the mixing time to be. And if you wanted to scale something up just by a factor of 100, you would need 2,000 times the power to stir the impellers to keep those vessels going. And so that's just not practical. And so what that means is that we're going to have different zones inside these bioreactors that have different conditions. That could be from the hydrostatic pressure, as I was just mentioning, or it can be from things like this, where certain cells get hot in a zone and deplete nutrients, and then the mixing takes a long time to even all of that out. And so one of the other pieces then is, as these environments are different, the cells are then going to respond to those environments differently. So you can imagine an easy one, which would be coupling oxygen discussion for a facultative organism. So if you've got an aerated zone, it can be doing respiration, generating lots of energy. It can take up a lot of nutrients, and it can do the chemical conversions rather efficiently. If, however, it goes into an anoxic zone, even if it's temporary, now the cell's going to shift to a fermentative metabolism. How much of its proteome is going to be reallocated to now adjust for a new environmental condition, even though 30 seconds later that cell may now be back in a well-mixed, well-aerated zone. And so these losses and changes in the proteome allocation can start to decrease the amount of nutrients that are available for ultimately becoming our products. And so this is one of the reasons you see performance in terms of titer and yields go down as we try to scale up these processes. So how do we study this? Well, the computational fluid dynamics are now being backed up by putting actual sensors, little floating softballs, that have different probes inside them into industrial reactors, and then you can map the timelines or the distribution of how long a particular spot in a reactor is at a particular condition, or what the lifetimes would be for a ball moving around in the vessel. And so with that, we can get distributions of environments, and we can start to study the organisms in bin groups of those. And so here I'm using dissolved oxygen as, again, one of the variables that we might be interested in. And so you might have a highly aerated, reduced aeration, and an oxygen-starved environment. And we can study the performance of our organisms in each of those cases, and then mathematically integrate, based on the distribution of those spaces in our real bioreactor, what the performance would look like. That, however, doesn't take into effect a lot of these hysteresis. So when the cell is going between these environments, there's additional wastes that wouldn't be modeled appropriately here. And so this is where the systems now come in with different types of bioreactors that we can build in the laboratory to simulate what's going on inside the actual industrial reactor. And so at the top right here, you see two bioreactors that are paired. So one could be set up in an aerated, one could be set up in a starved zone. And you constantly move the material between the two vessels. And so the cells are going to now experience those two zones in one combined batch system. You can do the same thing by putting in plates that are semi-permeable in a large bioreactor, and so the nutrient and the media exchange between these zones then can dictate what the conditions are, and you can put multiple layers of these plates to best simulate whatever your large-scale reactor is. And so now the challenge is we don't have that many large-scale reactors. We certainly don't have one in CABBI at this size, so we are going to have to simulate what we think the likely design would be in those spaces. The one challenge with these single systems is that you're just getting bulk average. You don't get to look at what's happening to the cells during these different types of stresses. And so this brings me to the system that I studied in Germany, where we couple, instead of two stirred tanks, we couple a tube, and we call that in chemical engineering a plug flow reactor. And so we have media that's flowing slowly through this column to the right of the stirred tank, and that could be where we introduce a stress. It could be a lack of new nutrients, so the cells would go through a starvation. I get the 2-minute warning here. And so then or it could be something where we add a pH starvation, a toxin starvation or challenge, and we can then monitor as the column goes up the different time exposures to those stresses and how the cell is responding, what proteome allocations are changing. And so to kind of go through this quickly, This is publications from the Tokors group that looked at an organism and how its stringent response goes up as the cells experience a carbon starvation in this plug flow reactor. It then looked at the functional genomics and identified sets of genes that are differentially upregulated during this starvation that aren't needed for their general performance. And then when we deleted those genes, and in this case they were flagella elements, things that were unnecessary for the bioproduction, they found that this reduced genome strain, or the streamlined strain, had a reduced maintenance energy, and that led to higher protein production in the form of GFP. And then when we turned this to a fatty alcohol or fatty acid producing strain of the same organism, we ended up being able to show that there's no difference in the laboratory. Right, so these bars here have no statistical significance. But when we moved them to this simulated industrial reactor, we got a 10% boost in performance. And so these are the things that we think are important to look at in terms of the CABBI yeasts. And so Shifang is going to have a poster over there, and she'll gladly talk to you about some of the work that she's been doing. We've now built one of these systems in Madison, and we're going to continue to scale these things up. Her first experiments were looking at some of the tau-producing strains, trying to simulate this one publication from a couple of years ago. We've managed to simulate the growth in the steered tank reactor correctly, and then some of the first hot off the presses data, we were able to now see that there's a complete, as you would expect, decrease in performance when we go to these plug flow reactors. And so now we're looking to see what's happening to these cells, and that's the future of these types of experiments. So I'll leave you with two kind of thoughts. One, that the environment inside these reactors is not homogeneous, and so we need to understand how our biocatalysts are going to perform, not just in the lab ideal environment, but also in this distribution of others. And then we're looking in the future to use this type of platform to do basic science, to understand how to build better biocatalysts that can be used for whatever product, but then also to help the groups here test and see what their strains are actually going to do before they go to commercial production. So with that, I'll take any questions if there's time.